Welcome to class. We are on day 3, so past the halfway point. Today, we're getting into a lot more really fun activities, I think. Starting to do read mapping, which is an important step for all sorts of different bioinformatic workflows, so that should be useful to everyone. So I want you to understand the general algorithm behind read mapping before we actually do it. So I'm going to talk about that. Then we're going to actually do it on the command line. And then I'm going to do some slides about experimental design. And that's really important to the success of your experiment. I'm going to try to convince you as we go through this course that everything you do up front as far as planning the experiment and getting high quality RNA and getting high quality libraries is just as important as everything you do on the back end for informatic analysis. Another really fun thing we're going to do today is visualize the reads and actually look at our read alignments using a viewer from the Broad Institute. So it should be pretty fun. Did anybody have any questions from yesterday? Yep. Just resize enter. Single word, hit enter. Yep. And it really does help if you're seeing that weird, like, it can happen if you're using nano as well, where all of a sudden the text looks like it's just doing weird things. You're like, oh, what's going on? Quit nano and hit resize. Or if your lines are wrapping over on themselves as you type. Just resize and enter. Yep. Mm-hmm. Regular sequencing. So deep sequencing, I wouldn't say that I know if there's an actual officially accepted cutoff, but deep sequencing, I would consider getting a lot of coverage of all of the different parts, all of the different genes that are being expressed in that tissue. So it's not, in my mind, when you're working with a large eukaryote and the ENCODE consortium, actually this will be in a lecture today, they recommend about 20 million reads, which I would say right now is probably about normal coverage. 80 to 100 million reads is deep sequencing. Like you're really fully sampling rare transcripts. And that's just their cutoff. It was developed for human and mice as a standard. If you're working with microbes, you're gonna need a lot less coverage for it to be considered quote unquote deep. So kind of, I think it depends on the organism of interest in the community that you're in, what's considered deep. Yeah. Nope, I think deep sequencing is just a way when you're writing an article to say that you think you've really sampled everything, even the most rare transcripts. Just a keyword to say that I've done a really in-depth, thorough job here, but a bit subjective. Yeah. Other questions? This whole middle part of the room is very quiet. You guys need to like develop some questions. No? Okay, so I'll start talking a little bit more about read mapping. Hopefully, get the slideshow. There they go. Okay. And I'll also talk about the GFF file format. So I wanted to give you a positive spin on today, like it's gonna be really fun, we're gonna map reads. The negative side of today is we're gonna be talking about a lot of really boring file formats. So you're just gonna have to try to stick with me on that and learn what we can. File formats are very boring, but they actually turn out to be incredibly useful when you're trying to actually do computational work. Most of the times because the file formats either are mislabeled or there's some discrepancy and you're the one who gets to go in and sort it out. So if you don't understand the formats, that can be tough. So we started out the course and did a little bit of BLAST. And BLAST is probably the first thing that most people learn when they're starting down the path of computational biology. And now we're going to be talking about aligning short reads. And I had mentioned this before, but I wanted to get a little more in depth. These are very different activities. And conceptually, they're kind of similar. You're taking two sequences and you're trying to figure out how similar they are. The BLAST and short read mappers are actually used for very different approaches. Alignment methods, they must have trade-offs. And depending on the application, you may want to make different trade-offs, which is why there's different pieces of software available and different approaches. For BLAST, what we're looking at, first of all, in comparison to short read mappers, BLAST is extremely slow, like much, much, much slower. And it was just not designed to do the same thing as a short read mapper. It's designed to really expect much longer sequences, over 100 base pairs, probably more like over 400 or 500 base pairs is when it really works well. But on the plus side, it can detect evolutionary relationships even after a significant loss of percent of identity. So we could take a human protein and a plant protein and potentially find homology. There may only be 30% difference, 30% remaining sequence identity, but BLAST is sensitive enough to detect that. Short read mappers, on the other hand, were developed specifically when next generation sequencing emerged because we had so much data and we needed to be able to deal with it. And so they're much faster and they're specifically designed to handle very short sequences. When next generation sequencing first really became commercialized, it was only about 25 base pair reads, so extremely short. So they're really designed to handle reads down to that size, but they're only going to find matches of 90% identity or more. You're not going to be mapping from human to mouse or from human to plant. You're going to be mapping human to human, possibly human to chimp. Like your evolutionary relationships are just conserved for what you can do as far as mapping short reads because they have to be such a high percent identity. So we have peach reads. We're going to be mapping them back to a peach genome. So it's a really good fit for a short read mapper because they should be very high percent identity. And the great thing about using a short read mapper is several tens of millions of reads can be mapped per hour per CPU, which is what we need when you have millions of reads. Also differently from BLAST, we saw that BLAST, when we ran it, it would give us many, many, many matches, starting from this is your best match, this is next best, all the way down to things that didn't look like they were particularly biologically significant. Short read mappers are not designed to do that. They're generally by default only going to output the best hit that they can find or the set of all hits that are equivalently good. So if the read maps to 4 or 5 locations in the genome and they're all equally good, they all score equally well, you're going to potentially get all of them back. And the reason why this is, the point of BLAST is often to find those kind of distant relationships between proteins or between sequences. But the point of a short read mapper is usually to find the origin in the reference genome. We've sequenced a peach, we have a peach genome, we want to know where did this read come from in the peach genome. It's just an intrinsically different question. And thus other regions, if they're lower percent identity, we don't care about them, they're generally discarded. So I mentioned some reads can be mapped uniquely to the reference, some reads map to multiple locations. Reads that map to multiple locations can be difficult to figure out what do you want to do with them? How do you want to handle them? Are they important for your downstream questions? In RNA-seq, if you're seeing a gene map to multiple locations, it's probably from a gene family member or part of a repetitive element. And if you don't know which gene they come from, can you use that data? Is that helpful for you? If you're looking for variants, where does that variant actually occur? So how do you deal with multiply mapped reads? A lot of people just throw them away. This is the default in most applications. Most short read mappers will give you the option to keep them, but you will have to turn that option on. Or it may, oh, that's actually not as true today as it was a few years ago. Most of short read mappers, depending on what software you choose, you should read the documentation and figure out what they're doing with multiply mapped reads. Are they putting them in your output file and tagging them as multiply mapped, or are they just not mapping them or not reporting them back? So just something to be aware of that's kind of going on behind the scenes that some people just aren't sure how that's being handled. You should go research it and try to figure out how is that being handled in my pipeline. However, short read mappers do have a myriad of different ways to try to avoid this scenario. Can we try to find a best alignment and really anchor that read to the correct origin in the genome? So the first thing you can do is, if you discard multiply mapped reads, if you have gene family members and they share regions of high similarity, you can discard the multi-mapped reads and then make the assumption that the multi-mapped reads would have mapped in the same ratio as the unmapped reads. Or not unmapped reads, the singly mapped reads is what I meant there. So what I'm trying to say there, let's say you have gene A and gene B, and the very first part of the protein is very unique. So you have 100 reads mapping to gene A and 10 reads mapping to gene B, just in the unique region. Then you have all of these reads that could map to gene A or gene B in the non-unique region. Well, what do you do with them? If you throw them away, it's going to make it look like the expression of your gene is much lower because you're just throwing away a bunch of your evidence. So what you do is you assume that those reads would have mapped in the 100 to 10 ratio that you saw from the unique region. So this is basically a way to do some statistical massaging of your data and avoid this problem of multi-mapped reads. So discard them and still have some accurate quantification of gene expression. As far as software mapping programs, the ones that actually use quality values when they're trying to pick the best alignments can do a better job of picking the real location that a read originates from. Maybe there is 1 or 2 bases that are different between those 2 regions. If you use the quality values of the bases, you can be more accurate in picking which region did this actually come from. But probably most importantly, if you're concerned about this, paired information is going to get you the furthest. Because if one read does not map uniquely, but its pair does, its pair can anchor it to the right location. And I talked about that a little bit yesterday. Would it help if I drew that? Does that make sense? Where like one's uniquely mapped and one's not. The uniquely mapped one tells you where the non-uniquely one has to go because you know how far apart they are. Yeah. Use the K means clustering on what type of data? Yeah, you could do something like that, looking at expression patterns and seeing how those play out using the uniquely mapped expression values, and then inferring what that means as far as the non-uniquely mapped reads. Yeah, there are a number of different clustering algorithms that do similar things to that. I could point you to some papers. But yeah, there's many interesting ways to statistically get out of situations where you're trying to figure out my reads are not uniquely mapped, or other situations. When you're doing, I think I'm thinking more of like when you have a transcript reference and you're trying to figure out which transcripts originate from the same gene. You can do some nearest means clustering for that. What type of motif are you looking for? It's a real short sequence, maybe 9 character long, so that's in there, then I'm looking across strains and so use a short reader when I use RNA sequence. So are you working with an organism that doesn't have a genome? And does it have a reference genome and NCBI? I would just look in the genome then. Because the RNA, unless you think that the RNA is going to reveal something that the genome doesn't. Does that make sense? Because the RNA is being transcribed from the DNA. So everything in the DNA, the RNA shouldn't necessarily tell you anything new, unless there's like some post-transcriptional modifications. But I wouldn't think that wouldn't be as big as like a whole 9 base pair insertion. So looking straight in the DNA for your motifs and figuring out if it's in a gene, I'd start with the genome in that case. Yeah. So yeah, decisions for the end user. Even when you're working within the short read mapper world, you can decide if you want it to be slower and more sensitive, or faster and less sensitive. Some programs actually have really nifty flags like -- sensitive or -- fast that kind of make these decisions for you. But others, you need to read the manual and figure out all the different parameters that you can adjust. And sometimes this just depends on, are you trying to map, is the individual U sequence quite genetically distant from the reference genome, or is it quite similar? Is your poster due tomorrow and you need it to run very fast? Or do you have plenty of time to let the sensitivity be really high and wait? Another decision that you're gonna have to make, how many mismatches are allowed for a read to be considered mapped? That's usually a parameter that you can set. So again, if you're just concerned about mapping more distantly, you might want to increase the number of mismatches that are allowed. That will make everything slower. If you have an incomplete or low quality reference, that could also influence your decision here. And the software will generally let you control how many matches to report, and if you should include multiple equally good matches in the output. But overall, because there's so many different programs out there, my recommendation is to explore the documentation and parameters for the software that you choose, and make sure it's doing what you think it's doing, because it's sometimes these things can be quite nuanced and you need to really understand it. For RNA-seq, the most common programs that I see are HiSat2 and STAR. HiSat2 is actually the newest version. It's kind of a rewrite of TopHat. So TopHat really dominated the market for a very, very long time. TopHat used an underlying aligner called Bowtie. So you might have heard of this tuxedo suite where they're all named after articles of clothing: Bowtie, TopHat, cufflinks, all that. So lots of people are still using TopHat, but you really need to stop using TopHat. If you go to the TopHat homepage, it says, we've rewritten it, we're no longer maintaining TopHat, it hasn't had any updates in over 3 or 4 years. Please move to HiSat2. So that's what I'm telling you, move to HiSat2. STAR has been around for quite some time. It's still being maintained. It's very fast. It's very accurate. And that's one we're going to use in this class. But there's many others, and your lab may be using something different, and that's potentially okay. If you get to make the choice of what software, pick something that's common in the literature. Your paper, you don't want a reviewer to question the legitimacy of your results because you've used something that they've never heard of, unless you can really defend it as, this is why I used it, and this is why it's the best for my problem. Good documentation. Don't get involved with software that is a black box and you don't know what it's doing, and some error comes along and you're not going to be able to solve it, and you don't know how it's handling multiply mapped reads. If the documentation isn't there, don't use it. You want something that's memory efficient and that runs relatively quickly. With the ACF especially, you've got 24-hour time limits on jobs, so if it can't complete in 24 hours, you're going to be in trouble. And usually in the literature, if something is really moving the field forward by being more memory efficient, that's going to get highlighted in the abstract of the paper. A responsive mailing list or help forum can be really great, and it may be, honestly, a really, really good mailing list that's been active for a number of years. You aren't even going to have to ask your question because someone else has already asked it and authors have already answered it. That's the best case scenario, right? Is you type in your error or you type in your question and Google says, hey, that already exists and the authors have already answered that question. So that's a huge advantage of having a responsive mailing list. And then if you are a unique problem arises and you're the person who gets to ask the question, if you can actually get somebody to respond to you, that's really nice. So I like to check for that before deciding to use a piece of software. Make sure it's maintained and that it's updated when bugs are found. So if you're looking at a piece of software and it hasn't been updated since 2012, I would say you should avoid it, especially because things do change, operating systems change. If the software isn't updated, there's no guarantee it will work. And also it may just be out of date and something that if you try to publish, your reviewer is going to say, well, why didn't you use this cool new thing that works way better? So we've run into that if you can't tell. So, just think about these things and try to pick something that makes sense on all of these different parameters. So what mappers do have in common, and we saw this with BLAST too when we ran make BLAST DB, is using this indexing strategy. So you want to transform part of the data or all of the data into a suitable form for fast searching. And this is very similar to having a book where you have a glossary in the back and you're able to look for instances of a short word and it'll tell you where to go in the whole sequence to find that word. Without indexing, the computer would have to scan the entire genome or the entire reference transcriptome every single time it was looking at a new read, and that would be really, really, really slow. And this is really similar, if you're just thinking computationally, this is really similar to how Google works. Like when you type a word into Google, it does not go research the entire internet. It does not go look at every page on the internet and say, hey, what do you know about the search term? And hey, what do you know about the search term? What they're doing is they're crawling the whole internet all of the time and creating this giant indexed database of all the keywords. So when you type in whatever your keywords are, they compare it to that database, find all the pages that they've previously looked at that have matches, and then send you to those pages. So that's intermediate step of indexing the data before, ahead of time, so that when you do a search, you can do it very quickly. Okay, so we are going to use another little kind of tiny toy example to try to figure out how this works, or demonstrate how it works. And this is our reference genome, 50 or 60 bases up there, I think. But in your mind, you're gonna imagine that that's 1 billion bases long. And you have a read, and this is quite a short read, but you're gonna imagine that you actually have 100 million of these reads. And they're actually more like 100 bases long instead of whatever that is, like 15 or 20. So this looks like kind of a weird, like when you're looking at it, you're like, oh, this seems like a lot of work for what we're doing. What you have to imagine is the reason why we do all of this is because you have to scale this up so massively. So you start with your reference and you start creating words. So in this case, I'm just pulling out all of the 6 base pair words one by one by one. So just each word that I can possibly find in the sequence, moving down 1 base at a time, and creating basically this list of new words. And in bioinformatics, you're often going to hear these also referred to as k-mers. So when you're reading the literature, you may see k-mer. This would be a 6-mer because it's always 6 bases, or a word size. And then you're gonna, in the case for this example, we're gonna alphabetize those words. So we created all the words, but they're all out of order. That's not really that useful to us. So we're gonna alphabetize them so that it's gonna be easy to find what we're looking for. And now we're gonna do essentially the same thing from the read. We're gonna take our read and we're gonna create all of the words, and then we're gonna take all of those words and put them in order. So if we have our reference word list and our read word list, we now have 2 things that are much easier to compare. So when we started, we just had a whole bunch of nucleotides in the reference and a whole bunch of nucleotides in the read. And how are we going to figure out where the read goes in the reference? Well, now we have something that's a little bit more of a tractable problem. We have a reference word list and we have a read word list, and we can just start going down and comparing them. And if you start comparing them, you will quickly realize that you do have words in common between the reference and the read. So I started with a reference sequence and a read that were almost identical. But why? So why would I only have 3 words in common? Why wouldn't I have more? Did anyone, and we can go back to so looking at that, and you can see this read was derived from the very front of that reference genome. Why don't I have more reads in common? I mean, more words in common, sorry. There's a SNP. So we have 1 base pair that's different. And it turns out, do you want to elaborate on that? Well, okay, so it doesn't impact a single word, right? It's every word that SNP occurs in no longer matches. So from that CTATAA, you know, that word has changed. You move down a single base, that word has changed. You move down a single base, that word has changed. So it impacts all of the words created upstream and downstream. So this is why it's important to have longer reads, because to find these words that actually match and anchor reads to the correct location, you need a certain number of base pairs that match exactly. And you need enough of them to indicate that this read probably goes to this location in the genome. So if I move forward, so even though this was like 90% identical, we're still only seeing a handful of words that match. But it's enough, and this is my alignment where I've drawn boxes around our words. So none of the words that contain the G or the T in this case matched, but the flanking words did match. So this would be enough information for a short read mapper to say, hey, this looks like a really promising area. This looks like this is probably where this read goes. And then to go in and try to figure out where are the SNPs, where are the insertions and deletions, and anchor it and do the full alignment. But basically, creating these word lists and comparing them is how a read lands on a part of the genome. And then the mapper can go in and sort out all the nuances of the little itty bitty differences and do the alignment. Does that make sense? And that's why read mappers are really fast. Okay, so this is pretty straightforward if you're mapping DNA to DNA. You've got another layer of complexity with RNA-seq, at least with a eukaryotic organism, right? Because we've got introns. And introns create these huge, massive gaps when you're comparing an RNA sequence read to the original genome. And you also have alternative splicing. So there's other gaps. So the gaps may not be consistent between different transcripts that originate from the same locus because you can have alternative splicing. So the first thing you're going to do to combat this is pick a read mapper specifically meant for RNA-seq. Don't pick a read mapper, don't pick like BWA. It's looking for DNA because it's not going to be designed to take into account introns and alternative splicing. It'll probably work sometimes, but it won't be as good as a mapper meant for RNA-seq. And the software will actually be able to take a file, a GFF3 file, which we're going to talk about in just a minute, to know where the genes are located and to automatically splice out the introns. So this is, if we were mapping straight to a genome, some reads would map perfectly on top of an exon, and that would be fine. But some reads are actually going to be split between the first exon and the second exon, or the second exon and the third exon. So when the read mapper is trying to find this, think about all of those words that it would not be able to use because of that massive gap. If instead the read mapper is aware of the introns and cannot consider them, all of a sudden those reads, it knows, yes, those reads are mapping to the same location. It's just those two exons are next to each other. So it's basically kind of pre-processing your genome to know where the introns are, if that makes sense. So pick a short story, pick a piece of software that understands genes. Not a big deal if you're using microbes. Those of you who are using microbes, don't worry about this. So now we get to another fun file format, the GFF, the generic feature format. This is actually quite an old file format. It's been around since at least the 90s, maybe earlier. But it's gone through a number of iterations. And it's moderately related to the GTF format, which stores the same type of data. So the big problem with this format, as I was saying earlier with FASTQ and FASTA, those are really hard standards that the whole world has adopted. Everybody's behind them. Everybody agrees on what they should look like. If you get a file from Japan or Europe or Illumina or PacBio, it is largely going to be formatted the way you expect. GFF is more the wild, wild west. You get a lot of strange variations on this format. My lab struggles with this a lot. We're trying to load data into our genome database. So there are some things online that try to solidify this into a bit of a more standard format, but just fair warning, you're probably going to see a lot of weird variations on it. So GFF3 was one of the original attempts to take GFF and make it quote unquote standard. But they kind of went a little overboard, I think, in trying to preserve backward compatibility. And so backward compatibility is basically you're trying to not break what's already out there, what's already out in the world. You want to add standards and you want moving forward to create files that are more compliant and more standard, but you want your old files to still work. And so sometimes that's a trade-off, right? By refusing to break the old standard, they had to actually leave the new standard so maybe a little too flexible. And so that's why you can still see a lot of different variations. But basically, what you can hope to see is a 9-column tab delimited plain text file. And I probably skipped over this part, but the point of this file, the point of a GFF is to describe where things are. So you have a genome and you want to know where a gene is. Within that gene, you want to know where the exon is. You could also describe repetitive elements. Where in my genome is a repetitive element? From this base to this base. And so this is an example of a line from a GFF3 file, and we're going to break it down. There's a lot of pieces here. So the first column is always going to be your chromosome or your source sequence. What is the background on which the feature is annotated? So for a genome project, you're generally going to see chromosome 1, scaffold 1239. You're going to see something that's like a big piece of the genome that's kind of the base of what you're trying to annotate. So that's always the first column. The next column is the source of the annotation. This could be, this was annotated by Maker. This was annotated by Ensemble. It may be the software that did the annotation. It could also be a database. So if you're downloading a genome from Vitosome, it might say the source of this annotation is Vitosome. If the person who made the GFF file has neglected to put a source of the annotation, the way you basically specify not applicable is to put a period. So a lot of the files I download, I end up seeing a period in that column. So it's not actually a required column. It has to have something. It has to have at least a period, but it doesn't have to have the source of the annotation. The next column is the feature type. So what are we annotating here? What are we looking for? Often it's a gene or an mRNA or an exon or a repetitive element. But that third column should specify what is being annotated. It must be a term or an accession from the sequence ontology. You guys will largely be working with these files being made by someone else. You won't necessarily be making them, so you don't have to worry about that. But basically, there's a controlled vocabulary out there, and you're supposed to pick a term from this vocabulary, not just make up whatever you think this feature should be called. Next is the start position with the sequences numbering starting at 1. So that's just saying like the first base of the first chromosome is numbered 1. The next base is number 2. The next base is number 3. And so in this case, we have a gene. It is starting at the 301st nucleotide of chromosome 1. The end position of the feature, same thing, just where this feature's end. So this gene stretches from the 301st base to the 2169th base of this chromosome. Next, there's a column that is supposed to hold a score. The score is not standardized. It has no actual definition other than if you have a score derived from some piece of software or just your personal gut feeling about the gene, I don't know, whatever you want to put in there, as long as it's a number, it's like technically compliant. But I just want to warn you, like when you see those scores, it can be hard to interpret unless you know what piece of software created those scores and how it created those scores. There's no actual standardization at this level. Like a GFF file can have any, like you can put any score there. There's no standardization of what that score means. Next, and you can just put a period if you don't want to put a score. Next, you have strands. So as we know, DNA, double helix, 2 strands. You need to specify if you're defining the gene as existing on the forward strand or the reverse strand. Repetitive elements, you might say, well, they're on both. So you can put a period there, but in general, it will specify if this is, you're annotating a thing that is functional in the forward strand or the reverse strand. Next is a phase column. This is an interesting column. It is here solely for the purposes of CDS features, which are coding sequences, which are, I will talk about in just a minute. They're a sub annotation of a gene, and they are used to indicate where the open reading frame starts. So it's a 0, 1, or a 2. A 0 indicates that the first base of the feature is the first base of the codon. 1 indicates the second base is the second base of the codon. So basically, this is just telling you what frame you're in. And it's only used, it gets its own column, but it's only used for CDS type features. Every other type of feature should have a period in that column. And then finally, the 9th column, attributes. And this is a theme that we'll start seeing a bit more of. Well, I guess it's more in variant call format files, but there's a bunch of bioinformatics data formats that kind of use this hey, there's an empty field, put whatever you want in it. And the way you can put things in this field is you have a tag value. You can have a set of tags and values. So in this case, we have ID equals whatever the ID is, name equals whatever the name is. You can have any other information you want to put in that, as long as it's a tag equaling a value with a semicolon in between. So it's kind of just this big flexible, you know, I have a junk drawer in the kitchen. This is like the junk field. I mean, there's usually useful information in there, but because there's no real standard format, you can just put lots of different types of stuff in there. And whoever created the file, sometimes you have to ask them, like, what do these tags mean? So that's a bit of an unfortunate part of the GFF3 specification, but there it is. But the most important part of the junk drawer, there's one thing in there that is very important, and that's the parent feature. So I've been very negative toward GFF files, but this is actually brilliant. This was like a really brilliant thing that someone came up with. And that is that when you're annotating a genome, it's very hierarchical. You have a chromosome, you have a gene. Inside that gene, it has exons, it has untranslated regions, it has introns, but all those things are associated with the gene. And so they created this parent tag. And basically, you're able to say, let's say you define a gene, you can then define an mRNA that's parent is this gene. You can then define an exon whose parent is this mRNA, and a coding region whose parent is this mRNA, and the UTRs and their parent is the mRNA. And this allows you to cluster all of these little different bits of annotation into, and know that they all belong to the same gene. So it doesn't, yeah, it's still a bit confusing, but it is kind of a nice way to understand annotation. And this became really, really important and useful when we realized that most genes are actually alternatively spliced. So this file can explain to you in a machine readable, computer readable way how alternative splicing in an organism works. So a gene can only have one parent, but an individual item like an mRNA can have, so a gene can have multiple children, and mRNA can only have one parent, if that makes sense. So this gene can be spliced in two different ways. So you can define two different mRNAs that have the same parent gene, and each has different exons that have a parent mRNA. So this is how you describe alternative splicing. And this is an example that I took from somewhere that has a gene that has different alternative splicings. So basically on CODTIG 123, we have a gene from 1,050 bases to 9,000 bases. Its ID is Eden. We've got a note saying it's protein kinase. Again, junk drawer, you can put whatever you want in there. And then this has 3 different mRNAs. And these mRNAs hold information on our understanding of how this gene is alternatively spliced. So it has multiple, in this case, they didn't define exons, they've defined CDSs, which is non-standard, but that's another story. But it can have 4 CDSs, it can have 3 CDSs, or it can have 3 CDSs with a slightly different location. So this is useful. This is why you need this file when you're doing RNA-seq, because otherwise, when you're trying to map reads back to a genome, you don't know all the different places that those reads could come from, or all of the different ways those little bits of the genome have been spliced together to make transcripts. So it's a boring file. It's not fun to deal with. They're often non-standard, and you have to go in and edit them and make them all, you know, work with your software program. But they're super necessary for this type of work. Any questions? Yes. Note equals protein kinase. What is the? So you can put, the GFF3 file format specification does not say what you have to put in this last column. You can put anything. And it turns out the different gene annotation programs put different things because the software authors just thought, hey, this is a piece of useful information. And different individual labs who are producing reference genomes might put different things. So in this case, somebody thought, hey, it would be really nice if I had a note about what this gene does. So they put a note equals protein kinase. That's not required. It is not something you will see in every GFF3 file. Someone else may decide to call it function equals protein kinase. They can name whatever tag and value pair they want. It's just a non-standardized part of the file that I can't tell you what it's going to look like because it's non-standard. We generally use the GFF that comes, so we've started using the breaker pipeline for gene annotation, and so there's maker and there's breaker. They're kind of, I think when breaker came along, that was like a play on words against the maker was the original genome annotation program. We just use what comes off of breaker. When we're downloading from NCBI even, these are very non-standard. The parent terms are not named the way that makes them identifiable in the database. You may have a FASTA file with a certain set of sequence IDs, and the GFF3 has a completely different set of sequence IDs. So we just are we're generally the cleanup crew as opposed to the producers of the file, which is why I have a deep love-hate relationship with these files. They drive us kind of nuts. Yeah. So, but good question. We don't tend to store protein functional information in these files. The biggest utility of this file, other than mapping, is for building JBrowse or other types of browsers. So I can show you that. Let's see what we have. Right, yeah. The important thing is to have a unique ID that matches your actual sequence IDs and to have the parents sorted out correctly, to have the right parents. Whether or not it has functional annotation there is not that important. And let me see if I can think of a genome that would be working here. Perhaps since this here has a JBrowse. Yeah, so here's a great example of we're just zoomed in on a gene, and here's some more examples. So when you're building these online visualizations, this is a GFF file specifying where to draw these boxes. It says, hey, you're looking at contig 1. We have a gene. There's an untranslated region, there's an exon, there's an intron, there's another exon. The way the software knows how to draw these little boxes is because it's reading that GFF file. If we didn't have that file, it wouldn't know where to draw the little boxes. And then we also produce GFF files to show a green ash. We've mapped the green ash genes onto the European ash genome because up until recently, we didn't have a green ash genome. So again, we built a GFF file to say, draw the boxes and put names associated with those boxes here on this contig at this location. So it's also an important, the reason why we build these files is also to make human intuitive visualizations. Yeah. Any questions about that? All right, next up, Matt's going to talk a little bit about a paper that my lab recently read about the STAR mapper and what can go wrong, where it works and where it doesn't work. Let me see if I can find his PowerPoint. Yeah. Do you know where he went? Okay, hold on. All right, we can take a 5-minute break. I'm sure he just ran to the bathroom or something. See you guys at 9:50. Oh, it's awful. What kind of? Awful. Well, it's mostly like we. It's not. It's usually complete and total failure. It's not like you get more like you're just anything that you can't even write up the file or decide on the file. But these are all not like or because it'll be like okay, so yeah, I need to come in and see your R1, page R2, page R3, and then you have a file that's named 1 3. So it's your file. I think it depends. Like what people's they've done animation yeah, you can use embedded. If I did that one with a recent job of like the MCPI, which is like absolutely all the genes will be named whatever they use sessions. You can't figure out what sequence goes and yeah, it's not good stuff. It needs a breaker and all this stuff around spit out what they're gonna name the gene on the baby. Or if they change the underlying genome. Oh yeah, yeah. It's a lot of that. That's also just like we just don't know what it's very confused. How I gave them a 5-minute break. 6 more minutes.
All right. Sorry about that.
Yeah, that's why it's nice to have on. We've been trying to add some of our J browsers some RNA coverage. And even nicer is if you have Instagram. Oh, that's great. Around us is my database forever like absolute there.
Ready to start?
I get 1 more minute.
All right. All right. So today I'm going to be giving a brief overview of a paper that is called The Fractured Landscape of RNA-seq Alignment, the Default in Our Stars. This is going to be a very brief overview. This is a chunky paper. There is a lot of crazy figures in there. It really goes into depth of how each of these alignment tools work, like the normalization methods they used. I'm just going to give a brief overview of some of the findings they found in terms of how these RNA-seq alignment tools work. So to start with, the most common method of quantifying gene expression is by mapping RNA-seq reads to a reference genome, which is what Meg just talked about. Then afterwards, you can count matched reads and determine differential gene expression from that, which is part of what we're doing later in the course. So definitive alignment is challenging because of factors such as gene splicing, short read sequences, which is what we work with, and paralogous sequences. The benchmarking tests that are used to analyze these new tools, they tend to focus more on the technical metrics. This group believes that they need to start focusing on the biological metrics as well. So their study is, they're hoping to assess alignment choices from a biological standpoint, which focuses on, in particular, they're focusing on the splice transcript alignment tool, or STAR. So this is some information on the data sets they used. One database they used was the Vivatis data set, which is a collection of 462 human lymphoblastoid cell line samples. One thing to note is the number of female to male lines. Female human genomes have 2 X chromosomes. Male genomes have a single Y chromosome and an X chromosome, and that may result in some differences in expression between the two. These are also available on the European Nucleotide Archive with their accession number right there. To assess the RNA-seq expression database, they used the GEMMA, ARCHES, and Recount2 database. GEMMA is a quality control database that used Bowtie2, which is out of date at this point. ARCHES used Callisto, which is another alignment tool, and Recount2 used the Rail RNA pipeline. For STAR, the study used version 2.4.2a. This is very important when you are citing something. There can be little differences in how a program works based on the version number. I've had this issue before in the past come up where one of the people reviewing our paper wasn't able to reproduce our results. So one of the things we had to do was go back and see what version we used. So this was done on the Vivatis dataset files using human genome version GRCH38.P2 and GenCode version 22. Following parameters were changed compared to STAR's default settings. They changed the minimum alignment score to between 0.55 and 0.99, and they allowed the number of mismatches to be between 0 and 9. To quantify expression levels, they used RNA-seq by expectation maximization, or RSEM. So I'm gonna just skip through all of the results because I tried having them on a slide, but there's just no way any of us can make out what was going on. There was just too much. So I'm going to summarize their findings right now. They had very helpful notice of alignment generally working well across all algorithms until it fails. So in particular, significant changes in the ability to report gene expression is tied to situations in which the precise sequence is required, such as XY paralogs, or when obtaining exact values, in this case, expressed versus not expressed. They've also found that on a purely technical sense, it's really difficult to pick a best practice due to the similarity of the outputs. This group recommends that to move past these technological comparisons and improve the biological assessment of both data sets and alignment tools. So rather than just looking at comparing how they run in terms of runtime, they need to start looking at how they handle different biological data. Their steps that they suggest are first and foremost, understanding what causes the differences in performances rather than focusing on obtaining good performance. So understanding these differences can more accurately identify where optimization methods result in trade-offs. And then knowing these trade-offs would allow for more improved decision-making in selecting a quote-unquote best tool. Their second step is to have a greater understanding that these metrics show clear signals precisely in areas with little variation in biological signal. This could be because the signal is too strong, such as sex differential expression, or too weak in the case of low expression. So, in short, RNA-seq is there's not a lot of biological understanding of the biological variation between the different alignment tools, and this is something that needs to be addressed going forward, and it's something to keep in mind with your data.
Is RNA-seq under the covers of the map? Is it used heuristics, or is it just straight statistical algorithm?
These programs I believe these programs use statistical analyses.
It's like that word thing that I was showing you. Like, imagine if there was exactly every word, you never find the line. So basically, there's a lot of making fashion. So that's a problem. And I don't know. Do you remember Matt in the paper gave some examples of regions where like genomic regions where RNA
Not the top of my head. I think that their main areas were just like differentiation between the sexes, and
I think there's also like the MHC very closely related to very difficult for certain members to do it. In Julie's project, we have a region of 6 genes that are canonically duplicated and are all really, really, really similar. And I mean, just annotating them. The first time, and I was actually constant the first time they discovered they were trying to annotate it, and it's less. It's very recent annotation. And there's some evolutionary divergence, but not a ton. And we know that with RNA sequence map, it's
That's very interesting. Usually with universities, you have confidence because it's a mathematical statistical guess. Universities, that's what the confidence is. In abundance, but it stays 7%. What's happening. But you make a great point. If you didn't have that, you're trying to do just a straight math algorithm.
yeah, and there are actually mathematical proofs that show how to obtain absolute perfect best alignment for a reused genome. Making things real trouble if it's only reads, right? So that's where we started. We were rarely having sequence data. This is a graduate student. One of the guys I work with, my PhD, his entire PhD was 4 years of sitting on and that was and he was super proud of that. And so he could work with algorithms that like literally compare one string against another string of absolute perfect because he had to run a gel, like sequencing back then. He had to run a gel for everything. So we give off a lot of the But if you're working in a region like NHC is the one I know from animal really all across here. Those regions were love and attention. There's a lot of things we're fine. There's some reasons. Yeah, you guys should consider reading that paper. It was an interesting paper. All right, now we get to get back on the computer. No more, you still get to listen to us talk, but it'll be more interactive at least. Okay, so we're gonna log in to the ACF. I am gonna go copy that Q sub command. And paste that in. That'll take about a minute. So how many people know what heuristic means? Anybody? I probably should have covered that. All right, you guys want to talk about heuristic? Here, Charles, you want to give a definition?
A heuristic is just a lot of times doesn't have any basis other than intuition.
Yeah, yeah. Do you want to comment on it, Ken?
The way I was taught we were doing AI machine learning is normal programming has if-ends, so it's a very logical branch. And there are times when that doesn't fit the situation we're trying to understand. So heuristic is if then if this happens, then maybe this is true, maybe this is true, and it's based on some discovery stuff. To get you closer to a solution that may not be a correct.
Right. Right. So for those of you who are trying to follow our earlier discussion but didn't really understand heuristic, that's a great, it is, it's kind of an approach to problem solving that's practical, maybe more that you're making some trade-offs. It's not 100% accurate. It's not a perfect mathematically defined, you're absolutely going to find the right answer, but it's maybe it's a way to do things a little bit faster and approach a problem practically, but maybe not perfectly. So a lot of, it occurred to me while we were waiting on these ACF jobs to launch, we should talk about that because this is a lot of how computational biology works now. Because of the amount of data we have, there's a lot of heuristic algorithms that we need to employ to process the data. So, okay. Did anyone have trouble with their Q sub? Who's still waiting? You're still waiting? Are you the only one? Okay. Oh, you're still waiting. Is it like the same people who were always waiting as yesterday? Oh, okay. I thought it was, yeah. I thought it was some personal bias the ACF has against you guys. But no, it appears that maybe it's just random. I don't know. Okay. So for those of us who have logged in, let's also CD to our directory. Cluster courses. Haven courses. Hopefully everything completed yesterday and you have got some trimmed FASTQ files hanging out in your directory. And also, to always do PWD and make sure that you're where you think you are. Say Luster Haven Courses, EPP 531, and your username. And uname -a tells me that I'm on a beacon node, not on a login node, which is good. If your QSub doesn't launch for 2 or 3 minutes, you can just use the login node. I did email Nick about the login problems yesterday, and he has not gotten back to me. So hopefully we'll hear something soon. I like to keep things really neat and tidy because a lot of these programs, especially like we're only dealing with 6 sequence files, but you can imagine if we were dealing with 100 sequence files and we did our quality check and our trimming and our mapping, and we did it all in one directory, you'd end up with thousands of files in one directory, and it gets very difficult to manage and find what you're looking for. So I like to try to put all of the results of one analysis into one folder. So if I come back later and I have a question about trimming, I have a question about mapping, I can find the files that I actually am interested in. So in that spirit, we're going to put all of our trim files into a directory. So I'm just going to call it skewer. I also like to make the name of the directory, just like the name of the files, make the name of the directory informative so you know what's in this directory. And I'm going to move all my trim files into that directory and all of my log files from skewer into that directory. And now I should have a much cleaner directory without all those files in it. And if I ls skewer, you can see they're all still in there. They're just hidden away in a folder. Okay, so very similar to what we talked about in the lecture and what we did with BLAST, we're going to do a 2-step workflow to map reads. The first one is to generate that genome index file for our reference sequence. We have the reference sequence in a folder that is accessible to the whole class called genome. Don't go into that folder, we'll just use it from where it is. But we have a GFF3 file and we have a FASTA file. I'm going to do a head on that GFF3 file so you can see it. Okay. So this is looking just like what we heard about in lecture. Our chromosome, in this case, the chromosome is PP01. That means peach, Prunus persica, chromosome 1. This annotation is from Phytosome. Phytosome only holds plant genomes, but they have been, the files have all been sanity checked. And when you download something from Phytosome, it will make sense, which is super nice. And you can see we have a gene followed by an mRNA. That mRNA has exons. These are our gene IDs. It has the parents set up. The parents are actually parents that exist in the file. So this looks like a pretty sane GFF3 file that we're going to be able to use to help us with our mapping. When creating the genome index files, the mapper is going to take both the genome and the GFF3. And that's so that when it's creating those words, it can create words that span those exon to exon boundaries and skip over the introns. Okay, so the program that we are going to be using is called STAR. So we can do module load star. And we are going to each create our own index inside of our home directory. So we're going to call that star IDX. And then this is the whole command. You can feel free to copy and paste. I am going to run through, yeah, I am going to copy and paste it, and then we will go through each and every one of these flags to talk about what they mean. I think this takes a few minutes, so we'll have plenty of time to go through the flags. Okay, so the first one is the run mode. So star can generate a genome or it can map reads. You need to tell it, I'm trying to do the first step. I'm trying to do the genome generation step. So that's the first thing you're going to tell it. The genome directory is where do you want your index files to go? You need to know where those index files are so that you can later specify on the mapping step where they are. In this case, we're just telling it, please put those index files into a folder in my current folder called star IDX. That's what the / means. In this folder, there is a folder called star IDX. Please put my genome directory files in there. Next, we have a flag that's going to specify where the FAFSA file is. This is the FAFSA file. It should hold all of the, in this case, we're only using chromosome 1 because we don't want to take everybody's time for hours on end waiting for the mapping. So we've only taken peach chromosome 1. But if you have multiple chromosomes or scaffolds, you would shove them all into one FAFSA file and give that entire FAFSA file to the genome generate program. Next, I have run thread n equals 4. This is just saying, hopefully to speed this up a little bit. I was testing it last night and it was a little bit slow. So I'm telling everybody to use 4 threads instead of 1. So you're actually asking the computer to run 4 processes in parallel to try to process this command. 2 threads can run on a single CPU. So if your computer has, most modern CPUs anyway, so if your computer has, let's say your computer has 2 CPUs, you could run up to 4 threads. Then a lot of the star options have these kind of really long names that start with SJDB. But one of them is SJDB GTF file, and that's where you want to specify your GFF3 file. This is SJDB GTF tag exon parent transcript. And basically, because I was talking about how GTF and GFF files are all kind of variable and a little bit different, it's asking you to explain to it how to group features. So in our GFF3 files, parent is the key tag term that is grouping features together to explain what a gene is. So you can just kind of memorize, if you have a GFF3 file, you use parent here. And overhang of 49. And that should generally be your read length, your maximum read length minus 1. So whatever, and you can find that out from your FASTQ or your trim output. Just take your maximum read length, subtract the number 1, and that's what you give to that parameter. And basically, that's just helping it to construct the splice junction database and map your reads more accurately. So telling the, at the indexing step, if you can go ahead and tell the software how long your reads are, it can do a better job with the indexing to make it more efficient. So that's a good thing to know. STAR has an extremely long manual that I will show you here. I think it's over 50 pages long. And it has so many options that it gets crazy overwhelming, but don't panic. It's got a lot of good information that will load. Here we go. And I'll show you, because what we've done in this class, and sometimes I think it's good and sometimes I think it's bad. We give you the commands, and like that's to build confidence. So you're like running the commands and you're getting results back and you feel confident. And the next step in your evolution as a computational biologist is you have to make those commands yourself. You have to read the manual and you have to figure out how to build all of the flags. And that can be like a scary next step because somebody hasn't written it all down for you. So I'm hoping that the material from this class will form a good basis that you can build on. You can look at the command from the class and just alter it to do what you want it to do. But at the end of the day, I guarantee you're going to have to go through these manuals and look up the flags and figure out what's going on. Most manuals will start with installation. If it has already been installed on ACF, yay, you can skip that whole section. And then it's going to talk about the basic workflow and the basic options. So even though the whole manual is 51 pages and it's probably worth skimming most of it, it's going to try to tell you what flags are you definitely going to need. What flags do you upfront definitely need to know? And these are all of the same flags that we're using in this command. And it gives you the same breakdown that I'm giving you on the wiki pages. So when I say pick a piece of software that has a good manual, this is what I mean. Like you want details, you want information, you want to be able to figure out what's going on. Particularly important for this class is somewhere in here, there's annotations in GFF format. So it says if your annotations are in GFF instead of GTF, this is what you need to do. So this is how we figured out as a lab when we were writing the lesson, that we need to put SJDBGTF tag exon parent transcript. Like, we don't have this memorized. Like, I don't want you to walk away from this thinking that like, oh, we just type this out on the command line every day and we totally have this memorized. We're digging through the manual, we're copying and pasting these flags. I mean, most of the stuff isn't named in any sort of memorable way. So as you are going off and doing this on your own, this is how you're going to find this type of information to build your own commands. Okay. Did most people get to the point of successfully having an indexed genome? Yep, nice. Yeah.
We want to go to our lab, something has arrived and it's very, very urgent. Can we do?
Okay, so this star mapping command is going to take about 10 minutes. So we're going to go ahead and get it running, and then we're going to talk about all of the flags, and then I've got a few slides on more file formats. I know everybody's excited. Okay, so I'm going to get that going. That's going to take a little while. So this is the star command for mapping the reads. In this case, we don't have to give it a run mode because the default run mode is mapping. Previously, we had to tell it the run mode was genome generate because that wasn't the default. But in this case, we don't have to tell it anything. When we do need to tell it where to find those index files, so we use a flag called genome dir, and say, go look in that star IDX folder that I just created to find those index files. We have to tell it where the read files are. You can put multiple read files in for a single mapping output. In this case, especially for our time constraints, we're gonna have a single read file. I'm gonna go ahead and have you guys try to use 4 threads, again, to see if we can speed this up a little bit. Tell it how you want to name the output. So we had our sample was named 8400. That's a meaningful name to us. We would like for our output files to also be named 8-400. And then this is probably the most important thing to talk about. By default, star is going to generate a SAM file. In this case, I'm going to let star go ahead and do some extra work and go ahead and generate a sorted BAM file for us. So this is going to save us about 3 additional command line steps, but it's going to be really convenient, as you're going to see in a minute. Did you guys have any question about this command? Okay. So now I'm going to talk about SAM and BAM formats and a format named CRAM. There's a new one. I have to go find the PowerPoint real quick. Okay, SAM format. This is the sequence alignment / map format. Whenever you map reads, this is the primary format that you're going to get your read mapping back in. This is standard, largely secure, appreciated worldwide, much better than GFF. More similar to like fast and FASTQ, where I can say this is in good shape, it's a good format, it's standard, lots of people use it. Lots of different pieces of software produce it, and it's pretty interoperable. There's a few little things more on the variant calling side where the SAM format gets a little confusing, but for our purposes, it's in great shape. It is a tab delimited plain text format, so it's human readable, which is great. And it was built specifically to store large nucleotide sequence alignments. It has the alignment of every single read in the format. It includes gaps, SNPs, and structural variations. So it includes all the details about how the read was aligned. It includes information about how reads are paired. It can store information about more than one alignment location in the genome. So if you have a SAM file, it can have reads mapped only to the best location, reads mapped to multiple locations, and it's very flexible. It stores quality values for the reads, and it stores information about duplication. So if reads are in multiple locations, it has that. The strength of the SAM file is that it's very flexible. It can store, as I mentioned, lots and lots of different kinds of information, but it's also fairly efficient. So it's useful for operations on very large sequences. Probably one of the things that made it a solid standard as soon as it emerged that everyone adopted is this extremely detailed documentation. There's really, the fewer questions you leave open about how data can be stored, the better the format is, right? Because then you don't have people all making different decisions. So that was a good thing that this format started with. And it comes with a software tool, or there is a software tool that you can use to manipulate SAM files, and it's called SAM Tools. We'll be seeing that. So the structure of a SAM file is a header. So the first, however many lines are needed to express the header, are at the top, and they always begin with an @ symbol. Below that, each line, there's one line for every read in the file. And those read lines do not start with an @ symbol. So that's how you can tell when the header has ended and the actual read mapping information begins. It's because it no longer starts with an @ symbol. So the header contains lots of information that I would call metadata. And metadata is data about data. It's not the data itself, it's the data about the data. So what version the SAM and BAM specification has undergone updates. So it says what version of the standard is being used. Is this file sorted? How was it sorted? What reference sequence was used? Any processing that's been done to the file that gets added to the header. The software version that was used to produce the file. So that means when you get a SAM file, even if you didn't produce it, even if you did not produce it, or if you did, you can go into the header and you can learn about when was it made, who made it, what software was used, is it sorted. So all the information is kind of carried along with the file, which is really nice. This is, however, it can take a little bit of effort to decode the header information. The SAM specification is in love with little short acronyms that make it a little bit of a pain to interpret. So @HG is always the first line. VN stands for version. SO stands for sort order. So this would be a version 1.5 SAM file sorted by coordinates. All of the reference sequences are specified with this @SQ. It gives you the sequence reference name and the sequence reference length. But again, they're using these like shorthands, SN and LN. So basically it means if you're actually trying to decipher the head of a SAM file, you have to go get the specification and go back and forth, and that's kind of a pain. But all information is there if you're patient enough. Okay, so once you get out of the header, we get to the actual alignment lines that specify how our reads were aligned. And similar to GFF3, it is a tab delimited plain text format with 12 or more columns. There are lots of columns. So we're gonna go through these really quick, just so if you look at a SAM file, you'll kind of know what's going on. In general, if you just did a header on, this would be like one line of a SAM file, and you might look at this and go, whoa, I have no idea what's going on here. And for the most part, that's fine. But I want you to be able to recognize a few key features. So we're gonna go through, the very first thing on the line is the query name. So this would be usually whatever unique set of alphanumeric symbols have been assigned to your query. The next field is called a flag. The flag has so much important information in it, but it's kind of hard to decipher. The easiest way, I'm not going to go through, there is a mathematical way to decipher it. I'm not going to go through that because I don't think it's worth the time in this class. But if you have a flag and you're trying to figure out what it means, you can go to this nifty website, type in the flag, and it'll just tell you what it means. And it usually means something like, this is a forward sequence from a paired-in sequencing experiment, and it's mapped in one location, and the next segment and the reverse strand is not mapped. So it has information like that. There's a whole bunch of, like, all of these different things get embedded in this one number. Again, unless you're having problems with paired end sequencing and you think something weird is going on, it's rare that you're going to be looking at this. But it's there. It encodes a lot of information. Next, it gives you the reference name. So the very, very first column is the sequence name. This is the reference name. So this individual read sequence has been mapped to this reference, usually a chromosome. It gives you the position. So the leftmost mapping position. In this case, it's mapped to 3,295,752 bases into your chromosome. You get a mapping quality. This is a standardized score that follows a defined mathematical algorithm. It's exactly the same as Fred. So it's exactly the same as the score that's given for base qualities. And it's basically just an estimate of what's the probability that this mapping position is wrong. So a map quality of 30, very good. We're assigning it a 99.9% probability of being correct. A map quality of 0 or 2 is very, very low. You would assume that this mapping position is highly likely to be incorrect. If you see a value of 255, that means that for whatever reason, the mapping quality value is missing. Next, they have this kind of shorthand to describe how the read is aligned. Again, this is not something you should ever normally care about. But for example, if you had 36 matching nucleotides, it would be coded 36M. If you had 8 nucleotides clipped off the beginning because they didn't align, and then 28 matches, it would be 8S28M. So again, these are things that you can look up, but probably don't care about. Yeah, in my normal class, I make everybody figure out how to read cigar strings, but we won't go through all of that. If you ever need it, you can look it up. There's a whole thing online about cigar strings. And so cigar is just, and I forget what cigar stands for. I think it's an acronym, but it's a way to encode these like complex read mappings into a very, very short set of letters and numbers. They look like nonsense, but if you're a computer, it's very meaningful. Yeah. So basically, all of these parts of the alignment are being encoded as letters and numbers. The next field is giving you information if it's really useful if you have paired end sequences. And it's telling you, if this is a forward read, it's telling you where the reverse read maps. And if it's a reverse read, it's telling you where the forward read maps. And this is just so that you can quickly and easily assess if your reads are mapping in the same location, as you would expect if they're paired. It also tells you the template length. So for paired end sequencing, again, how long was your original template? Then you get the entire sequence of the read is embedded in the SAM file and the quality values of the read. So this takes every single piece of information that was in that FASTQ file and re-embeds it into the SAM file. And then you have, again, this like junk drawer situation where the last field, you can just put all kinds of random information. And I have some examples. And this varies, like all of this stuff varies by the mapping program. So this is not actually going to be in the SAM format specification. If you're trying to figure out what this stuff means, you have to go back to your mapping program usually to figure it out. But it might have information like the number of best hits that were found for this read, the number of suboptimal hits, the number of gap opens or extensions, edit distance to the reference. So yeah, all sorts of different like little pieces of information the mapping program might care about just gets shoved into this last template. So when you do a head on a SAM file, what are the things that you're really looking for? At the top, you should see a few lines with @ symbols. Sometimes there are headerless SAMs that have no header lines, but usually your SAM files will have some type of information at the top. You may see something about what mapping program produced it, what reference genome was used, and then for the individual reads, you should see at a minimum, you should be able to identify read name, gonna start everything off, and you should see sequences and quality values. So if you're just trying to figure out what is this format of this file, my collaborator just sent me a .text, and I just want you to be able to look at a file and say, is this a FASTQ? Is this a FASTA? Is this a GFF? Is this a SAM? These are kind of the key features that you're going to use to figure out what you're looking at. BAM format is the binary version of SAM. It is basically taking the SAM and squeezing it way down into a much smaller file that is only readable by computers. This is really useful because SAM files are giant. They're huge. They will take out, we have SAM files in my lab that take up like 100 gigs. They're huge. And it just depends on how much sequence data and alignment data you're cramming into this one file. So basically, SAM tools, the software package, allows you to squeeze all of that down into a much smaller version of SAM. It's using a pretty clever compression algorithm that's quite common in Linux and computer science. It's a type of zip format. You've probably had zip files on your computer. It's a kind of a fancy version of that. They are bigger than gzip files, but they are actually able to be used for random access. So you can actually still do computational tasks using a BAM file, which is useful. But they're still up to 75% smaller in size than a SAM file, so that's great. But they're not readable by people. So if you do a head on a BAM file, it's gonna look like total gobbledygook. I mean, before this class, you may have also said that this looks like gobbledygook, but indeed this actually is structured, whereas this is what real gobbledygook looks like. So hopefully you'll be able to see the difference as we move forward in the lesson. There is a new format. It's been around for a while. I see it very rarely. But occasionally I'll run into it in a few software programs. It's called CRAM. It was supposed to be like the better, smaller, more awesome BAM. It hasn't really taken off the way that the software authors, I think, hoped it would. It's even smaller and more efficient than BAM, but like I said, most software programs don't really use it. And I think one of the things that has made it confusing is there's different types of CRAM. So we're not going to use that in this class. You're probably not going to run into it. I just want you to know that it exists in case you run into it. And we're going to talk a little bit, we'll do a couple of quick little demos in the class. But the main piece of software you're going to use to manipulate SAM and BAM files is called SAM tools. It's part of this bigger suite called HTS-lib. If you work on variant calling, SNPs, and indels, you're going to also get really familiar with BCF tools. We don't cover that in this class, but it's part of the same kind of suite of software packages. For our purposes, all we care about are SAM tools. That's what you can use to read and write SAM and BAM files and to convert between them. So if you have a SAM, you can convert it to a BAM. If you have BAM, you can convert it back to a SAM. You can also do indexing. We're going to see that. You can look at SAM and BAM files. You can edit them. So lots of super useful tools. All of the manuals and versions of SAM tools can be found at htslib.org. Any questions about our suite of new file formats? SAM, BAM, maybe not too much CRAM, mostly SAM and BAM. So hopefully if yours finished successfully, it should say finished successfully, and then you should have your very first BAM file. Who's actually got a BAM file? Okay, not good many people. You guys have a BAM file?
It's still running.
So the next part with SAM tools, if you don't end up actually executing any of this, it's not going to prevent you from moving forward. We're just going to play around with SAM tools a little bit. Okay, so let's go through some of these output files. Like some of the other software programs we've seen, you get a lot of different output files, some of which are very useful. The log.out file, this one, it has a lot of detailed information about the run. It is not generally that helpful, but we'll check it out anyway. I'm going to use Nano this time. This is a text editor. I don't think we've seen it since Monday, but when you open the file, it's basically a little text editor in your terminal. It's going to allow you to scroll down and look at the file. So if you do keep it, it has some useful information like the version of star that was used. So when you're writing your manuscript and you need to put the version in, you can go find that. Run mode, and it'll have some information about what parameters you use to run the software. But for the most part, most of this is pretty useless. So let's get out of there. The progress.out file, if you're mapping a really big file and you're trying to check on the progress, it's a file that's just being regularly updated with information about how many reads have been mapped. So we've already mapped them all, so this is not useful at the moment. But if 5 minutes in, you had looked and you would see, okay, I've mapped 1,721,008 reads, and you know that your actual number of reads is 3 million, you could say, okay, it's about 50% done. So this can just be helpful if you're trying to figure out when the job's going to complete. And it does give you some, it's kind of telling you how it's going, like what are the % of my reads being mapped uniquely? What is the length of those reads? So if you're looking at mapping that's going to take like 5 hours, and you look 10 minutes in and you see the 0% of your reads are mapping, you might say, maybe I'm mapping the wrong file to the wrong reference, or you can use it to go ahead and kill a run and say something's not right here and start over. Okay, I'm gonna go Control X to get out of there. And then the most helpful file is the log.final.out. So this is the one that has actually all of the information that you really care about. So first you can see that the mapping run took about 9 minutes. It was mapping 22.49 million reads per hour, so that's pretty good. Tells you the number of input reads. Back here, the average input read length, the uniquely mapped reads. So 91% of our reads mapped to a unique location on the Peach genome. That's actually pretty high. That's really good. When they mapped, almost the full length mapped, which is great. Gives you some information about splicing. Again, because we're doing RNA-seq, it's not surprising that a lot of our reads were spliced. We had a very low mismatch rate, a low deletion rate. So this is a great example of Illumina, right? Like the error profile for Illumina is very much SNPs, not insertions, deletions. And so what we're seeing, and whether this is real biological variation or actual or sequencing error is tough to say at this point. We are seeing about 2% of our bases are mismatched, so it looks like a SNP, whereas like 0.05 and 0.03 have insertions and deletions. So that's just a very characteristic Illumina error profile. About 8% of our reads map to multiple locations. Again, I'm going to take a guess that this is largely gene family members, areas of the genome where even though they're coding and they're being transcribed, they look very identical or very similar. And so the mapping algorithm is unable to figure out where they go. Do they go to one place or to the other place? And Shelley might be able to correct me if I'm wrong. I think STAR outputs multi-mapping locations. Yes. So right. So tomorrow we'll deal with those multi-mapped reads statistically, and in this class we're just going to throw them away. But there are smarter ways to deal with them. But in this case, if reads that map to multiple locations have definitely been output in the SAM file, and all of those locations are in the SAM file. We had a very low % of reads mismapped or unmapped. like essentially almost none. A few were unmapped because they were too short. That probably just means they were too short to find a unique location. No chimeric reads, that's a good sign. So yeah, so very useful output file, and that's the .final.out log file. So of the 3 files, the one I'm usually opening is this one. And could, so I think it's when you have 2 pieces of the genome that have been somehow recombined and been sequenced through. So if half of the read mapped to chromosome 1 and half of the read mapped to chromosome 8, you would probably flag that as likely a chimera. Like something has gone wrong, and some kind of recombination. I think it can be an indication of library problems. I've never actually seen a problem with the chimeric reads like showing up. Yeah. Control X gets you out. So yeah, so we asked the program to go ahead and give us a sorted by coordinate BAM file. BAM files can be sorted in 2 ways. The first way is sort by coordinate. So put the reads in order from like the ones that start at chromosome 1, position 1, chromosome 1, position 2, chromosome 1, position 3. And that makes it easier for a visualization program to draw regions because it can get all of the reads mapped to a region very efficiently. The other way to sort a file is alphabetically, essentially just to sort the reads by read name. Sorting by coordinate is 99% of what you normally want to do just for downstream applications. So that's what we asked it to do. But you can ask it to do unsorted, or I think it does sort by name. But we went ahead and asked STAR to do sort by coordinate, which is great, except we can't look at the SAM file. We can prove to ourselves that we can't look at the SAM file by doing a head on it. Oh, let me see what I've got going on here. Yep, so the lesson is wrong. I have an extra dot here that doesn't need to be there. So yeah, if I do a head, you definitely can see this is a binary file. This is a file meant to be read by a computer, not by a human being. So what if we really actually wanted to look at our reads and to see what our SAM file looks like? We can go ahead and go find that SAM tools software. Module load SAM tools. And then the SAM tools has many, many different types of subfunctions. So the first thing you always tell SAM tools is what you want to do. And I want to view my BAM file. To view in SAM tools, the word, it's kind of misnamed. Like I wish it weren't named view because that's a little confusing. But the view function allows you to convert between SAM and BAM. It also allows you to convert between SAM and CRAM and BAM and CRAM, but we're not going to worry about that. And to do some filtering as well. So it does a lot more than just view, but it's called view. We'll say SAM tools view. We want to keep the header. That's what the .h says. And then we're going to give it the BAM file name. So the only parameter we're going to give it is keep the header, and here's the file I want you to work on. We're going to output that into a SAM file. So I'm just going to rename it out.sam. And that's probably going to take a little while. In the meantime, let's go look at the SAM tools manual. Trust me, you will, if you end up doing a lot of this in your research, you will become good friends with SAM tools. So it's kind of nice. They have a few workflows that I have used in some of my classes for teaching. Just walking you through mapping reads and doing variant calls. They're also still pushing CRAM. Again, I don't know many software packages that use it, but here are the manuals. There's also how-tos. Those we can go ahead and click on specifications. So this is what is a SAM file? What meets the definition of a SAM file? So if you need to go through, you can and click through. There's a lot behind the SAM file specification. Yeah, there's a lot of information. So we're not going to worry about that. Just want to point it out in case you ever need it. What we're going to look at is the manual. So for SAM tools, each time a new version of SAM tools is released, it gets a new manual. And SAM tools has made real changes in how the tool runs on the command line based on different versions. The ACF is pretty woefully out of date. It's in 1.3.1. The current most modern version of the tool is 1.8. I guarantee you there are big 1.9. There are big differences between 1.3.1 and 1.9. But fortunately, we can still go back and look at the module. At some point, one of us will probably remember to email the ACF and ask them to update it, but it's been a couple years since I've remembered to do that. So this is how you would figure out how to use SAM tools. It tells you the view command, SAM tools view, can do all of these different things, and here are all of the different flags. It also has a sort command. So if you did SAM tools sort, you can sort files. Index, we're gonna see that a little later. You keep going down and there's just lots and lots and lots of different things. SAM tools is a big piece of software that does a lot of different stuff. So just pointing out where you can learn more about that. Okay, so hopefully if your SAM tools ran, you should now have a SAM file. And let's do a head on that. So if I scroll up, we have our header lines, which tell us a little bit about what's going on. It's a version 1.4 SAM file. It is sorted by coordinate. Our only reference sequence is PP01. And we use star. It gives us some information about the aligner. So again, if you ever get a SAM file and you're trying to figure out how the alignments were generated, it's going to have some information. It even has the whole user command line, which is pretty nifty. As soon as those lines are over, that means the header is over and we start getting into real reads. So we can see that we have our read name. It's mapped to the peach chromosome 1, 31,000 bases in, and we can see we have our full read and our read quality all embedded in here. So all that information is in this one file. Let's also do an ls/lh, and we can see the SAM file is 767 megabytes. So despite using a rather small data set, we have a very large file. The same file, when it has been compressed, is only 133 megs. That is still big, but it is much less big. So that is much better. And this is the reason we generally store files in BAM and delete the SAM, because they're just too big. It's going to eat up all your storage. You're paying for storage. Even if it's cheap, you might as well not pay more than you have to. So we're going to leave files laying around, leave them laying around in BAM format and delete those SAMs. Actually, as long as we're here, let's delete the SAMs just to make sure we don't overrun our project directory space. Okay, so I am going to let you guys feel free to work in pairs or small groups. Figure out how you would write a SAM tools command to convert to BAM. So we've converted from BAM to SAM. What if you needed to go back from SAM to BAM? So I want you guys to work together a little bit, go through the specification and try to come up with the command line that you think would work for that. I'll give you, let's say like 10 minutes. So, or we can round up. We'll go until 11 a.m. So if you need to take a quick break while you do that. But definitely get together and work with other people, it'll help. Yes, keep the header, and when you have a solution, write it on the board. own wild card. But yeah. Yeah, she owns that. A lot of group work. Everybody better go put their answers on the board. I will make this the homework. You guys all play along. So we already went from Sam to Sam, and now we're going to go to Oscar. How do you hear Sam? Just write your command. What will the command look like? What was your well, I used to use the operating. So yeah, so now I can. Eight times. Yeah.
Yeah, no, that's
I think it got rid of it. My new band trial is 30 minutes. It was too.
yeah.
Which is interesting. Is that more efficient? Yeah. Maybe the star is building as it goes. No, I think it's still the same. tried to run it yet? Any thoughts on flag to use? You don't use anymore. It won't hurt anything. That was one of the changes we made. You just don't need that symbol. You don't automatically do it. But if you put the capital S in there, yeah. So what we were talking about over here is you do that. Maybe you can make it smaller, the band star. Why? Why is it smaller? Yours is the same? Yeah, I mean, it would have to be, right? Like, that's what you're you went back to the same again. There's like a more efficient. Oh, I wonder if they've made the whole compression algorithm more efficient on sample stars just using an older something. Right. Yeah, there's no way to really interpret what's going on with the band file. Yeah. Okay. Anybody still working on the problem? Yes, that's everybody. So everybody use the lowercase b as the flag. And that's how you go the other direction instead of bam, 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 bam. And I have guesses only for why. Extra credit to anybody who can figure it out. Here's what's nice. You email them.
So both of those are binary formatted.
SAM is a text file.
So I'm looking at that, but you said extra credit. Like the two files.
So they're binary. They both convert back to the exact same SAM. Smaller size, you create the BAM samples of star. I'm guessing it's something different between star and how it does. Or I don't know if star whole sample. I think this is a much bigger issue. You're getting like a third of the file. It's a massively different size file. And that's what I'm thinking. The question is, I don't know how star does question. It's a column sample, maybe column conversion of sample, maybe I don't know. Mysteries. But the important thing is both BAMs convert back to the same SAM. So the information is still in there. There's no information loss, which is what we care about. Yeah, that was a fun little side activity. So I have put the loop for you guys to, if you are scaling up and doing this on your own reads. I don't want you guys to actually execute this though, because it takes 10 minutes per pile. It would take a full 60 minutes, maybe a little longer, for the loop to run. So unless you're just interested in letting it run in the background while we do other things, I would instead try copying the files. So I've made a directory called BAM files and put everything in there. So hopefully, I'm not sure if I got the permissions right, but if you copy BAM file star BAM to your current directory, you too have 6 BAM files. Did that work? Did anyone get a permission error? Oh, great. If it works for you, we know it's going to work for everybody because you're the one with all the permissions issues. So we should be really good to go now. And my job was just killed. which I guess is good timing because I was done. Okay. All right, we're moving on. Actually, I'd prefer to get the visualization stuff done today. So let's go ahead and move on to IGB, and I'll do the transcriptome project design tomorrow morning when we get here. It'll be a fun way to start the day. So nix that part, and let's move on to visualization, which Matt can do.
All right. So for my slides, I like to go through what we're going to do before we run it, and then we can go back and run through it together. So today I'm going to be talking about how you can visualize the results of an alignment such as STAR. We're going to be working with IGV, or the Integrative Genomics Viewer. It is my personal favorite tool for visualizing gene mapping. So after you perform your alignment, you may be interested in contextualizing your results with your genome of interest. In this case, we can see where our reads of interest align on our genome. And we can also include mapping information such as, we can also include like the proteins, where they begin and end, so you can contextualize this read is probably related with this protein. IGV is a popular tool. It is available in many formats. The one we are going to use is a Java-based desktop application, which I will show you how to download. But you can also use it on the web, and you can use it integrated into web pages. But just like when we ran BLAST, one of the benefits of running BLAST on the command line was that we can use our own references. That's the same deal with IGV. We can upload our own genomes onto IGV and run it with whatever genomes we have that aren't necessarily available through IGV's website. So it can view large integrated genomic data sets, and it also supports a wide range of data types. Most importantly, for our purposes, it includes BAM format. So we already talked about how STAR and how SAM files are really large and BAM files are compressed. IGV is a program that recommends using the BAM format when uploading sequence alignments maps. However, it has a few requirements. And this is what Meg was talking about earlier with why you want to sort your data sets by the coordinates rather than by the names. So it must be sorted by the coordinates, and it must also have something called an index file, which we'll get to in a little bit. So thankfully, we've already produced the sorted BAM file directly through running STAR. So that first requirement has been met. So now we need to produce an index file. So an index file, if you look at certain books, like a textbook, you'll have an index at the end, and that will tell you where a certain subject is. An index file works much the same way. An index file will basically, it's a companion file to the original BAM file that has all of the chromosome data, all the coordinates for where the chromosome begins and ends. And programs like IGV want an index file because then it can use it to more easily jump from one index to the next. So this is just reviewing how the SAM output works. So it's the immediate output of STAR, if we did not use the BAM options that we did, would not be ready for use in IGV. So the first step is to run SAMTools view and SAMTools sort together. Now, obviously, we didn't have to do that with this study, but in the past, that was how I learned how to index a file, because even though star gives you that option, not every alignment tool uses that option as well. I'll sometimes use something called BWA for my alignments, and that doesn't have the option to immediately convert into a sorted BAM file. So knowing that you can use SAM tools to convert to BAM and then sort the BAM file is very useful. So now we have our sorted SAM file, or sorted binary SAM file. Then you run SAM tools index to create a complementary file containing each of the genome coordinates. So this is what we talk about when we say scaling up. For the purposes of this lab, I'm just going to have you run SAM tools index on the same file, that 8-400 file that we initially started with. But you really don't need any other options when running SAM tools index. You just give it the file you want to index and then run it. The next step will be downloading IGV. I don't think, has any of you already downloaded IGV? Okay. So if you haven't, we'll go over the way to download it. So you just go to the download page and you select the app corresponding to computer's OS. So if you have a Windows, you'll select IGV for Windows. If you have a Mac, IGV for Mac. If you have Linux, IGV for Linux. So this is a review of methods of getting files from one computer to the next. So as with Meg, I'm going to be using the SCP, which is a version of this copy command or CP that allow you to copy from one server to another. There's also rsync, which is a more advanced utility than SCP. It can also allow for one-way transfer. So if you have a file on your computer that's really large and you want it on your server, but you don't want it saved on your computer, you can have delete source files once it's through downloading. For right now, we're sticking with SCP. Finally, Globus. So once again, this is an overview of how we run SCP. So the first option in SCP will always be what files you want to copy. In this case, we're copying from the server, so we put all the information for the ACF server here. That @ make sure that it connects the username with the server that you're connecting, and then the colon after the server name says within that server, this is the directory we want to copy from. So in this example, we're copying the FASTA file. We're going to need 4 files in order to get our data onto IGV. The FASTA file for the chromosomes, the GFF file, and the BAM file and the index file we made. So when you first load IGV, you're going to see that it's going to have something like the human genome preloaded. We obviously don't have anything to do with the human genome, so we're going to need to load our own file. So at the top of the menu, you have the genomes option. The first option is load genome from file. This is how we're going to load our chromosome. So once you select load genome from file, it'll give you this option menu, and you select the P-Persica FASTA file. Once it does this, it also creates an index file for the FASTA file, which works much the same way as the index file for the BAM file. And if we had more than one chromosome in this FASTA file, we'd be able to use that drop-down menu right here to switch between chromosomes. You could also use the search tool here to do the same thing if there are just too many chromosomes. Furthermore, if you have to switch to a different genome, you can go back to the Prunus persica genome by using that drop-down menu in the top left corner. If I were to click this on IGV, it would show all the genomes that I've loaded. In the case of when you freshly install it, it's just going to be the default genomes like the human genome. The next step, now that we've loaded the genome, we need to add the BAM files. This is simple. You just do load from file, you select your BAM file, and it will add it. It'll add 2 tracks corresponding to each. It'll add 2 tracks for the BAM file. This is the coverage track. You can't see it right now because it's zoomed out, but it will show you how many times a read matches with the segment of your genome. And then your second track is the alignment track, which shows you where each read aligns with the genome. So to zoom in, you have this bar at the top right corner, and you can click that to get into a location. There are many other options to zoom. You can also zoom in by clicking and dragging underneath the base pair count before the coverage track begins. So for example, if I were to click and zoom here, it would zoom into this location. You load the GFF file in the same way. This adds a new set of track, which is the annotation track. This gives you, and it might be hard to see here because of how the auto formatting set up, but this blue line right here represents the coding sequences from the GFF file. So it'll tell you where each exon begins and ends, and you'll be able to zoom in and look at where on your genome, where in the reads really match up with one of these genes. So I threw a lot at you. Now we're going to really try to understand what each of these tracks mean. So the coverage track, as you can see here, it's a bar graph illustrating how many reads overlap with the reference genome. You'll also notice that there are a lot of different colors. So the gray bar indicates that all of the overlapping reads match with the reference genome. Any other color represents a SNP, single nucleotide polymorphism. So, but these different colors all mean something. Blue represents cytosine, orange represents guanine, green, that really bright green, represents adenine, and red is thymine. If you see, for example, here, there are 2 colors here. That indicates heterozygosity. One of those colors is likely the reference genome. The other is the SNP. So in heterozygosity, you have a species where one set of the genomes is what you would expect from the reference genome, and the other set of breeds have a different map. They have a SNP. Next are the alignment tracks. So each read that aligns to the genome is represented by a gray track. If it's white, that indicates that you have a low quality read. And generally, the reason for that is that using transparency, IGV will de-emphasize reads of low quality. SNPs are color-coded, as we saw earlier. They follow the same format of green responding to adenine, blue responding to cytosine. You also have insertions and deletions. So they're represented by a purple I for an insertion or a black dash for a deletion. When you zoom in closer to the region with an insertion or deletion, they will also have a number, and that number will tell you how many bases have been inserted or deleted. I'm going to show you all of that in the next slide. If you see a black rectangle just underneath the coverage track, this indicates something called downsampling. IGV uses a lot of memory as it is, but it tries to optimize itself by not using as much. One way of doing this is through downsampling. It won't show all of the reads in the alignment track that align to the genome. It'll only show the most interesting. So these reads right here are aligning over a long distance, but they are white rather than gray. See, all of these are gray. Those are low coverage, low quality reads. This is an example of downsampling. So there are when you look at the coverage map, it says that there were 2,000 reads that align to this part of the genome. When you hover over the black dash, which I which I really tried to capture the message that it sends, but it just wouldn't let me because it's a pop-up menu that IGV uses. It says that I removed 1,846 reads from the alignment track. And finally, you have your insertions and your deletions. And what that little incident prevented me from showing you is that when you have when you zoomed in a ways away from the alignments, you'll only see a purple eye or a black dash. Once you zoom into the alignment, you start getting more information, such as, in this case, in the insertions, you have an insertion of 2 base pairs. In deletions, you have this deletion of 6 base pairs. Final thing I want to talk about is saving and reloading a session. So IGV can take a while to set up, and let's say you have some other project you're working on with IGV. Rather than having to reload everything and remember everything that you uploaded, you can save your session. So you go to the file and select Save Session. This will save your current session and everything that you've done with this session and allow you to reopen it at a later date. This is in a format called Extensible Markup Language, or XML. And I've seen XML used for a lot of different processes. Blast can save output as XMLs, which we can use to load on our website. The purpose of XMLs here is that it saves all the information of the genome that's been used, all of the BAM files that have been loaded, all of the protein files that are loaded as well. So to summarize everything, IGV is a tool used to visualize genome alignments. I've shown you how to index your file, download them to your personal computer, and load the genome, alignment, and protein files onto IGV. And using this, you're able to view what reads cover the majority of the genome, what bases are different from your reference genome, and contextualize the areas of high coverage with genome annotation. Anyone have any questions?
So now, like, Sam and Dan,
Well, in terms of the star file that you upload, they really recommend using BAM file. I haven't tried using a SAM file just because they're so large. I don't want them to mess up my computer. So now that I've gone over everything, we're going to go over everything again. And this time I'll walk you through it. So I've run SAM tools indexing on the login server without any issue. So if you don't want to do the Q sub, it shouldn't cause any problems. But if it does, then I'll take the blame for that because I told you all to do that. So go to the file, go to your star folder. And now Meg mentioned that she likes to save each of her files with a name that says, when you go back years later, it says that'll say, this is what we did here. I'd like to take it a step further and put a number in front of each of the steps that I've run so I can remember what order I ran it in. Now, you don't have to make any changes to the directory. That's just the way I did it. And when you're loading the data onto your computer, just keep that in mind that my file system was named differently. So since we're only going to be running the indexing on 8-400, this is the format that you want to run it with. Also keep in mind that because the star output that Meg provided doesn't have a period between 400 and alignment, to ignore that. Just remove the period whenever you're running indexing on your own. Has everyone finished indexing, or is anyone running into any issues? Everyone ready? And one of those programs that will show you nothing if everything goes well. That's what I was talking about when I make my directories, I use a number in the part order I ran it into so that when I look back at it, this one we're all going to be doing. And if data mover 1 is not working for you, you can find one of the other data movers. Copy the version that's on your directory already. Okay. So that's exactly what you said. File within your Did anyone else have any permissions issues with downloading the genome file? I think data mover 1 has been announced in data mover 1. So, okay. What data mover file are you using? Switch to 2. I have an issue with data mover. one. Ryan had an issue with Allison. You always need to have your destination.
I'm good. I'm good.
So first you need to and then you load the GFF file. You can load the file once. Download that from the PCM. You just need the diagnosis that will say, do this for GFF, and then also put it in the file. Yeah, you got it. Doesn't give you a feature. It's one of those programs that will have without really giving you features. Now, copy use a CPU. That's down. Mm-hmm.
That's interesting. So you have to
There's a lot of people sometimes that apply this idea. I think everyone applies because the state is
very good. So
it's very
yeah, makes sense. So
If you look at the reference statement, so I think what it's doing is that it's not really kind of because they're still within the same chromosome, but it's stopping, it's starting at one point and it reminds me of this is like the yes, they're in different so each one does correspond to
to say it's going to say how which is a safe section. Okay, there's the X. So then later on I can do a file load
that's where load the TFF file. So where the first term T chart. Really good at that. That's what.
It still seems
Down, but file. Loading yeah. So that's
The PDF it actually shows up. Yeah.
Here's your market.
So if you go to that generic drop down, okay, the first thing you do I was checking my email, like I was talking to like this big email.
So I went over
I sat down as an
three SCPs that flag those for all three SCPs that you have to run, copy the FASTA file, the FF file, and the BAM file.
Okay. He can probably help you more that point you where those are. I think they're in the lab.
They are. They're in the genome folder. Now, you can run. It says data move for one because that's the first data move. So you gotta use that. Lab implementation. That's the guy. We'll go back.
Yeah. They're there. How are you doing?
On this one.
Okay. If you guys take a few extra days on the same work, that is not a big deal. But it's okay if I like not eat your organization, don't panic. And if you want to come to office hours or wait until tomorrow, we'll keep working on it. We can do that.
Oh, just says like yes, because you use it.
Okay. Did you select the DAI file? Just do the BAM file. Don't worry about the BAI. It's going to find the BAI without you having to do anything. It's there. It's there. Actually, so if you right click without the BAI, right click on that one and just repair. I just forgot twice. Okay, now if you zoom in, you should start seeing alignments, which you can do again with the plus button. Yay! Alignments! And look at all of those reads mapping right on top of two axes of that gene. Okay, screenshot, almost done. Yay. You can also zoom in and look at more things. You don't just screenshot it and give up, but it's
Make sure you place username and then change that into issues. Probably because you're using it so much.
sometimes it's not super efficient and files that might crash on a lot. And sometimes, and this may not be really nice to involve in it, but but yeah, like as far as your programmer, you need to come away or you can file report also doesn't look like. Oh, it's big.
Star.
Yeah.
There's no green.
Yeah, Road is one of the
So that's the
I mean, I think it's well, they
After so space, after star. You always have to discuss conflict problem two.
There's like lots of puzzles. Yeah.
Yeah, that's the actual.
Yeah, and it's hard to run on a laptop. We're in good shape, but we're not that way. We have a big machine that's going down with the server. Portals, set up a portal, run a web browser, get the doctor or something, one other thing about it. So frustrating if you're not going to do it. If you can run IDB on the server, then the lag time that you press a button. You press another button. That's the same as in lightweight field. We get our next $3 million stuff.
On your pizza factories, yeah. Now, those band files, file. Then we load the AI model. It knows it's the and it uses that as the desktop. It's part of the there you go, drag. So these are the assignment is defined as a routine that is aligned. Find groups that are aligned to the curve. This way you look. Something like that. Does that make any sense? Is there anything you want me to touch on?
All right, guys, we're not going to introduce any more new material. You're welcome to hang around if you need more help with IGB. I noticed that the course surveys are now open. I recommend you don't take the survey until Friday so you can experience more of the course. But I'll remind you on Friday. We'll be getting an email if you haven't already about that. And yeah, let me know if you have any questions. Thank you.